Hello. All right. So in this video, we're going to be discussing eukaryotic cells. And as you can see, this is just part one. This is what a traditional eukaryotic cell diagram would look like. And as you can see, there's a lot going on. So I have broken up talking about eukaryotic cells into two videos. So let's first talk about what eukaryotic cells are. Remember that eukaryotic cells make up the eukarya domain. So when we're looking at the tree of life, there are three domains. The bacteria and the archaea are the first two. They are prokaryotic cells, are made up of prokaryotic cells. And then eukarya are named because they are made up of eukaryotic cells. And there are tons of different types of organisms that are made up of eukaryotic cells, a very diverse group. There's different shapes, there's different sizes. Some of these organisms are going to be unicellular, meaning they're only made up of one cell. But then some of these organisms are going to be multicellular. So we're thinking things like plants, animals, fungi, going to be made up of multiple eukaryotic cells. But all of them are together in that one domain of life because they're made up of eukaryotic cells. So in this video, we're going to talk about some of the components of a eukaryotic cell. And the first one that lots of people think about when you think of a eukaryotic cell is, of course, the nucleus. The nucleus is where the DNA is stored for the cell. It is made up of two layers of phospholipids. Well, actually, two layers of phospholipid bilayer. So technically four phospholipid layers. One phospholipid bilayer, which is shown right here, and then a second phospholipid bilayer. So there's two phospholipid bilayers surrounding the nucleus. There are these pores, we call them nuclear pores, as you can see here, that allow things to go in and out of the nucleus. So that's obviously really convenient. And then the inside of the nucleus is called the nucleoplasm. So kind of like how we call the inside of a cell the cytoplasm, the nucleoplasm is the inside of the nucleus. And in the nucleoplasm, there are two main things that are found in there. The chromatin, which is our DNA, and chromatin is just a fancy word for condensed DNA. So when DNA, the molecule is condensed, we call it chromatin. And finally, the nucleolus, which is the dark spot in the center of the nucleus. This is where a portion of the ribosomes are made. So if you remember, ribosomes are organelles, these small little hamburger-shaped organelles that produce protein for the cell. Now, where do those ribosomes come from? Well, a portion of it, not all of the ribosome, but a portion of the ribosome is made in the nucleus. So nucleolus specifically. So there's that. All right, outside of the nucleus, there is this other organelle called the endoplasmic reticulum, and there's a lot of production here. Things are being made. In the cell, there are a lot of things that need to be made. Proteins, carbohydrates, lipids, all of that. And so the endoplasmic reticulum is the main organelle where basically all of this production is happening. And you can actually see the endoplasmic reticulum in the previous slide. This is all endoplasmic reticulum, and it is very closely related to the nucleus. They work together. It is also made up of a phospholipid membrane. There's different sacs, tubules, layers, as you can see here. Some of it is patterned like this. Some of it has this kind of pattern, but that's what the endoplasmic reticulum is this kind of organelle made up of phospholipid membrane and all different kinds of shapes. And again, it is usually located attached to and near the nucleus because they work together to produce stuff for the cell. So there are two main parts to the endoplasmic reticulum. There's the rough endoplasmic reticulum, which contains ribosomes that are embedded on the surface. So as you can see, this is the rough ER. And there are these little red dots. Those are our ribosomes. They are literally stuck to the surface of the ER. And this is where those ribosomes are going to be making tons of protein, and that ER is going to be collecting all of this protein that's being made and kind of organizing them. And then the smooth ER, so attached to the rough ER, there is an extension of it called the smooth ER. There are no ribosomes here, which is why we call it the smooth ER. It doesn't have these freckles, basically. And in the smooth ER, we're going to be producing carbohydrates, lipids, hormones, all of that other stuff. So rough ER is where protein production is. Smooth ER is where everything else is made. And that is pretty much the job of the ER. So just think of it as like the manufacturing center for the cell. Now, once all of these things are manufactured, they need to be distributed and shipped packaged, etc. And that is a job of the Golgi apparatus. So the Golgi apparatus takes these items, anything, proteins, lipids, carbohydrates, whatever, these items that are made by the ER, they're going to be transported via a vesicle. So a vesicle is just a small bubble made up of membrane. And that vesicle is going to travel to the Golgi apparatus. And then the job of the Golgi apparatus is to sort it, tag it, package it, distribute it. Basically, the Golgi figures out where is it, what is this thing, and where is it supposed to go. And that is what the Golgi does. Some of these things need to be transported maybe back to the nucleus or to the other side of the cell or outside of the cell. So the Golgi just kind of determines where everything goes in the cell. Okay, and so as you can see, the nucleus kind of tells the ER what to do. The ER makes that stuff, and then the Golgi packages and ships it. And those are kind of the three main components. There are some other components to the cell that are really important, and those are going to be your peroxisomes and lysosomes. They are going to be our disposal center. Peroxisomes, specifically, they are these small vesicles, so they're made of membrane, a really tiny bubble, and they have ways to detoxify the cell. So whether it's some kind of toxin or anything like that, they're going to be getting rid of these toxic compounds out of the cell. And similarly, lysosomes, same thing. They're made up of a vesicle made of membrane, so it's a membrane bubble. And in these organelles, there are these digestive enzymes that break down any kind of unwanted material. Lysosomes are really important for our immune system because our white blood cells have tons of lysosomes, so that when the white blood cell, for example, finds a bacteria, it can destroy that bacteria using a lysosome. So that's just one example, but lysosomes are used in general to just break down anything that's unwanted. So for example, if a protein was made by the cell, but it's not functioning properly, the lysosome will break that down. So anything really that is unwanted is disposed of via the lysosome. So those are some two smaller organelles that are also really important when it comes to cell function. So that's all for this video. As you can tell, there's a lot going on in eukaryotic cells. I have this picture here showing all of the main organelles and their function. As you've noticed, we've only gone through about half of them, so kind of this half of the diagram. And so in the second video, part 2, we're going to go over the other half of the organelles that are found. Hopefully this video was helpful. I will see you in the next video.